So today we are going to talk about genetic screens, or what we call gene hunt as well. And in the same context, we'll talk about a very interesting concept in the mutant screen, which is called complementation. How we analyze the mutants, the ones which we discover, the ones which we have based on the phenotypes. But before we go into details of today's lecture, let's have a recap of what we did in the last lecture. And my computer is stuck. Yeah, okay. So far, what we have gone through is we have gone through detailed analysis of chromosomal theory of inheritance by mapping genes onto chromosomes. And we also talked about recombination, because when we map genes, we cannot map genes or we cannot develop a genetic map unless we know the concept of recombination. We did dihybrid crosses to map genes, and then we calculated the map unit distances. And also what was most interesting last time was the 3-point test cross, where we learned that if we are dealing with 3 different loci, which are A, B, and C, so how do we know it's A, B, C, or it's B, A, and C, or A, C, and B? And we covered great deal in last lecture, and also we had assignments given to you separately. Regarding molecular mechanism of recombination, we said this will be covered in 216 or in separate lectures. Today's lecture is basically, I'll leave you a few simple problems, and these are you are going to map genes on chromosomes, on autosomes. But before we go there, I would just like to highlight how to map genes on X chromosomes. Finally, we'll come to today's lecture, which is about gene discovery, gene hunt, or in genetic analysis, we call this genetic screen. And then in the context of genetic screen, we'll talk about complementation and allelic interactions. The non-allelic interactions we will talk in the next lecture. Okay? All this is part of our genetic screens. So this is a problem for mapping genes on X chromosome. This is just a quick, because we did not talk about how to map genes on X chromosome. It's not different. It's rather easy than mapping genes on autosomes, because you have to just look at the males in the first generation, just males in the first generation, and you can calculate recombinants, because male do not have a second X, and that will give you phenotype as well. For example, we have 2 recessive traits, cross-veinless and white, both lying on the X. So we have wild type fly, cross-veinless plus, we have cross-veinless mutant males, we have white-eyed males. So we just set up a cross, a female, which is white, cross it with cross-veinless male. In F1, due to a recombination between cross-veinless and white, we will get specific phenotypes. And if you cross F1 female with cross-veinless males, in the next generation you get the numbers. Idea is basically because we want to see the recombination during meiosis. And that recombination will tell us how much is the distance, how many centimorgans they are apart from each other on X. So this is meiotic recombination basically is being shown. If you have, this chromosome, which is basically this one, and then this chromosome, which is this one, if they are at a reasonable distance, uh, after recombination, so in next generation, we'll get these. We can easily predict the gametes based on phenotypes. You calculate the numbers. As usual, the biggest ones are the parentals, small ones are the recombinants, and I have this big rectangles, this big rectangle, because I want you to calculate and send as a assignment, little assignment. This is quick one. I think less than 5 minutes, you will get the answer. So the map distance between cross veinless and white. How much? From these numbers. Then mapping on autosomes. Let's say we last time looked at vestigial black, cinnabar. So today we give you an example of vestigial and short on the same chromosome. These are the wild types. So homozygous recessive vestigial short. You get F1 heterozygous. This is the tester male. Okay, tester male. These are the possibilities of recombinations. Of course, the non-recombinants, which are the parentals, the non-parentals. You have these numbers between vestigial and short. What is the distance between vestigial and short? You go and calculate similarly short and cinnabar. Going through crosses, you get the recombinants. So what is the distance between short and cinnabar? And then if you want to see the distance between all three, you got the the progeny of trihybrid cross. You can see these big numbers, small numbers, etc. You have to find the recombinant classes between different markers, and then tell us the distance as well as the gene order between cinnabar, short, and vestigial. Clear? So this is what is quick recap what we did last time. Let's now start today's lecture. And as I promised, we will talk in great detail about the discovery of genes, or what we refer to as genetic screens. Before we delve into the theory or the practices of these genetic screens, how they are done, let me tell you why do we do this. And here is wild type fruit fly, and you can see beautiful eyes, very beautiful bilateral symmetry, antennas, then the whole olfactory system, proboscis, etc. And as a scientist, you are interested in, let's say, eye development, how the beautiful eye structure develops. Or, this is the adult fly, this one. And the fly body is divided into 3 main segments. We call them head region, thorax, and this is the abdomen. And each segment is divided, like you see this, the fly body is basically segmented. And you can trace this back. So this is after 10 days when it hatches. The the foundations of these segmented body plans, they are actually laid down in early embryo. Few hours old embryo shows this segmented body plan. And you may be interested in understanding how the body axis or body plan is laid down. You may wonder why we are interested in fly body plan or segmentation. As I talked to you last time, so as a scientist, you always try to study a fundamental question in a simple system, and then you go to more complex systems because the fundamental principles of life are same. The genetic code is same. So let's say the ones who are interested in such a segmented body plan, if you are interested in getting answers to your question, someone may be interested in eye development, how eye develops. So taking genetics approach, what we do, we mutagenize. Remember I said we create mutants. And what are mutants? Our ideal mutant is a viable fly, a living fly, viable fly, which has defects in eye development, and rest of the body is fine. It's fully normal, rest of the body. The ones who are interested in understanding the segmented body plan, they found this mutant, for example. This is a normal fly. Look here, this is a leg. These are legs are coming in here. These are the legs. Normally, these are in normal fly, these are the legs. You can see here. Nothing onto the head region. But now you see legs emerging from the head region. These are the legs. Okay. So as a geneticist, you are successful here. You created a mutant which is alive, but it has 3 pairs of legs, but these 3 pairs of legs, which are normally present in the second segment of the body, they also appeared in the head region. And through this phenotype, you can now go and try to see what gene I have been able to mutate. What is the gene responsible for leg development precisely here always and not here? Because in normal flies, legs are not on the head. This is another classical mutant. This is my favorite one, for example. You see the adult fly has 2 wings. Both the wings are attached to the second thoracic segment. So this is the thorax. And so thorax is also further divided into 3 segments, T1, T2, T3, and wings are attached to T2. But thorax is normally fused. You cannot see them segmented like this. So due to change in morphology or due to mutation, due to genetic lesion, due to process of mutagenesis, you can see now the T2, the thoracic segment, is duplicated. This fly has 2 thorax, and classically we call this bithorax. Okay, mutant, bithorax phenotype. And there are 2 wings, 2 pairs of wings. These flies are unfit, they cannot survive long. It's very difficult to maintain them because they cannot fly, they cannot mate, they cannot feed properly. So this is the background for conducting a genetic screen, why we conduct a genetic screen. because we are interested in a fundamental question. We may be interested in amino acid biosynthesis, because we all know DNA is decoded into mRNA and mRNA is translated into proteins, and proteins are the functional molecules. If the building blocks of proteins, which are amino acids, if they are not available, organisms cannot survive. From where amino acids come? How amino acids are synthesized? That could be a simple question. And you have to bring answer to that. As a geneticist, you will go and create it depends which organism you're using. People have done these screens normally in the yeast, very successful screens. So what we do in genetic screens, for example, let's say here we are dealing with our amino acid biosynthesis screen. We are having, yeast. We have wild type yeast growing. We will mutagenize the wild type yeast. How we will mutagenize? There are special mutagens. Mutagens. So agents that induces mutations in DNA. And we see, all the wild type, they die, our mutants somehow survive. We call this mutant selection assay. You have to have very intelligent and smart assay in which you can select your mutant, because there will be so many wild type which may have gotten mutation due to treatment with mutagen, but they may not be of interest to you. They may not be in some amino acid biosynthetic pathway. There may be some other genes which does not disrupt amino acid synthesis. So you don't want to deal with them. So you have to select your mutant. And then what you do after looking at the phenotype of your mutants, you analyze if your mutant is dominant, mutant phenotype is dominant, or it is recessive. Once you finalize this, then you map the gene on chromosome. And once you map the gene on chromosome, you then clone gene. Okay. This approach is called forward genetics. Why? Because you have no idea which gene is involved in amino acid biosynthesis. Any gene in 32 chromosomes in the yeast may be any gene lying on the 32 chromosomes could be involved in amino acid biosynthesis. You have no idea which one is. So you are shooting in the dark, and then if you get it, you still don't know which is the gene. You characterize the mutant, and then finally you clone the gene and characterize the protein, its function within the cell, and you get all the answers about amino acid biosynthesis. Or you did this screen, you characterize whether this mutation, this classical mutation is called antennapedia. The phenotype was called antennapedia. So, or this one was called bithorax phenotype. So you have to determine whether this phenotype is dominant, recessive, on which chromosome it is. Is it on chromosome 2, chromosome 3, on chromosome X of the fly? Once you map the chromosome, then you map gene on chromosome, and finally clone the gene and characterize why I have legs on the head region, why normally legs are not present on the head. So it's a beautiful system. So you have to be intelligent which mutagen are you going to use, and there are certain considerations you have to keep in mind. We'll talk about this thing in detail in a few lectures from now. Then the dose of the mutagen, how much dose you are going to give to the living organism to create mutations. And a rule of thumb is that, you give a dose on which 50% of the wild type survive and 50% are dying. What are the kinds of mutagens? For example, you have UV radiations. Then there are chemicals used in microbes, nitrous acid and nitrosaguanidine. Then we have the EMS, MMS, which are alkylating agents. And more recently, we have transposons as well. We will talk in great detail about these mutagens in a dedicated lecture on forward genetics. Okay. So how do you get? So you got the answer. How do you get these mutants? You take wild type flies, these ones, you treat them, or you take bacteria, you take yeast, whichever model system you're working with, you treat them with these mutagens, and then you select for the phenotypes of your interest. Let's now take example of yeast, a simple model system, a eukaryotic system. It's a unicellular organism. Normally in eukaryotes don't have unicellular, but we call this lower eukaryote. This is the yeast which you also add in the pizzas. So remember, yeast does not reproduce sexually. They don't have males and females. Their mating types are called met A and met alpha. These are genes within the yeast on the same locus next to each other. And if this one is expressing, the yeast is called met A. If this is silent and this one is active, it is called met alpha. Why it is important? Because it's always met A cross to met alpha. It will never be 2 met alpha or 2 met A. They will never mate. Similarly, met A cross to met A, they will also reject each other. It's a simple organism with many chromosomes. So haploid is 16 chromosomes. How it reproduces, let's say this is the diploid yeast. Diploid yeast 2N is 32 chromosomes. It always divides asexually by budding. Just a bud emerges from the mother cell. And let's say you have now, when it undergoes mitosis, normal mitosis, so a mother will produce, so this was 2N, it will produce 2N if it underwent mitosis. Okay. However, if it undergoes meiosis, then it is going to produce, the spores, and from one mating type, one diploid, you will end up because as I said, these are like met A, met alpha. This is the diploid state. Okay, so you will have if G is a hypothetical gene, okay, after meiosis, one of the haploid will be carrying gene, and you treated this yeast with mutagen, so mutation, and due to mutation now you have a phenotype. The other, after meiosis, the other haploid will be G. They will be always met A and met alpha. Okay, 2 met A's and 2 met alphas after meiosis. 4 gametes, not gametes. Gametes are basically produced in sexually reproducing organisms. This is an asexual one. So we'll say there will be 2 met A types and met alpha types and 2 met A types. You can mate them, you can provide the pheromones and mix them together. They will mate and produce again 2N diploid, which is heterozygous. So both haploids and diploids are isomorphic. They look morphologically, they look same. And if you have haploid and it's a viable, it will give you phenotype. If it has dominant mutation in diploid also, it will give you phenotype if it is dominant. So by crossing, so you can cross this one, this haploid, observe its phenotype, cross it back to wild type. If the phenotype is dominant, then you say my mutation is actually dominant. If it is the phenotype disappears and wild type dominates, you say your mutation is recessive. Same things like we do in higher eukaryotes. Now, why we use yeast? Yeast is actually, how should I say, yeast genetics is even more well characterized as compared to fruit flies. And the biochemical tools and genetic tools, they are as good as fruit flies. However, the advantage is, this yeast divides in simply 2 hours. So one cell will go into 2 cells in simply 2 hours. In flies, you go in 10 hours, in 10 days. Also, growing yeast is much more simpler. You take salts, minerals, glucose, a carbon source, the minimal media, add amino acids, nucleotides, and yeast is good to go. You take, test tube, you take flask, you take 100 ml media, this growth media, you throw a few colonies or 1 colony of yeast, you come after 24 hours and you will see a type culture, millions of yeast cells there. Also, being unicellular, it's easy to understand the synthesis of complicated molecules because the analysis is easy in unicellular. But yeast has proved to be very useful in understanding, the fundamental transcription, basic transcription. How RNA pol2 comes, binds onto promoter, what are general transcription factors, how DNA damage takes place. You name any fundamental biological question in molecular biology, and its genetics and biochemistry had been very well studied in genetics. Now, I give you a simple example of amino acid biosynthesis. For example, here, we are talking about histidine biosynthesis. And, an amino acid does not come through magic all of a sudden. There's a biochemical pathway. Let's say a metabolite A is converted into B, and this conversion is due to one gene. Then B needs to be converted into C, another gene is involved, or another protein, not gene, protein product, which means gene products. C goes to D, and then eventually we have the histidine. Okay, so this constitutes a biochemical pathway. And finally, the histidines, they go in protein synthesis when ribosomes are decoding the messenger RNAs. What do? You took yeast, You mutagenized it with, let's say, a chemical called EMS or MMS or whatever mutagen you used. And then you grew this yeast after mutagenesis. You mix the yeast culture, millions yeast cells with this chemical in media, and then you grow the mutants onto plates like this, individual colonies. Now remember, this plate contains complete media. Complete media means everything is there: salts, minerals, glucose, all the amino acids, etc. So every colony which we spotted here, it grows happily, even though there will be some ones who do not make histidine. And you should know, if there's one amino acid is missing out of 20, how many genes will be impacted? So give me answer. If you have mutation in histidine biosynthesis and a mutant cannot synthesize its own histidine, what will happen to the living organism? How many genes will be impacted, or gene products? G. If an organism lacks the ability to make an amino acid called histidine, how much impact on the life of an organism will be there, and why?
Cells won't be able to synthesize some essential proteins.
Yes, it will not be able to. Very good. It will not be able to synthesize essential proteins, which means if it lacks histidine, it will be most likely, dead. Because not just one genes will be fine. Just one gene is mutated, which is the histidine synthesis gene. But since histidine is not there, transcription is taking place of all the genes, but translation is defective because histidine is not there and proteins are disturbed. So organism will be dead. So that's why in the first instance, you grow your mutants onto plate carrying histidine as well, so that it can utilize histidine from the media.
So I had a question which is somewhat related to this.
Pardon?
I had a question which is somewhat related to this.
Yeah, please go ahead.
So I wanted to ask कि mitotic recombination हमने बात की है के बारे में हमने बात की थी in the last class, and that may cause a change in the genome of the resultant cell, right? But will that change, for example, in a multicellular organism? अभी तो हम yeast की बात कर रहे हैं, yeast के but in a multicellular organism, will that change? For example, उस change के से जो है हमारी recessive gene जो है वो homozygous recessive हो गई है उस recombination की वजह से, that that change in the gene will not be expressed in the phenotype, right? Because वो सिर्फ एक cell while growing.
Yes, you're right. In a multicellular organism, yes, you're right.
Okay, but in a unicellular organism it will be expressed. अगर हम yeast की बात करें, for example, yeast के अंदर let's say mitotic recombination हो जाती है,
it will.
That should be expressed. Okay.
It will. Good. Very good.
Okay. Thank you, sir.
So now this is the but we are interested in histidine phenotype. Histidine phenotype means delta means deletion, or means it organism which is not able to synthesize histidine. So what we do then, we prepare replica plates. We take another plate which contain minimal media, this one now, only salts, minerals, glucose, as well as amino acids, but minus histidine. Okay, it lacks histidine. And then next plate where we have minimal media plus histidine. Now what is going to happen? We are going to take a stem, like let's say this is a stem. Okay, we put a velveteen cloth on this, and this is a plate जिसमें आप bacteria भी grow करते हैं BIO100 में. You come with velveteen cloth with the stem, you put on this plate, and that plate is which one? This one. This one, the wild type one. Okay, you go with the stem and then you stamp this onto minimal media with minus histidine, and then again stamp onto minimal media with histidine. What you will get now, you will get the phenotype: growth on minimal media and growth on minimal plus histidine. Wild type, since wild type can synthesize its own histidine, so wild type will grow on any media, whether it has histidine or whether it does not have. But your mutant, if it is in histidine biosynthesis pathway, let's say this gene is mutated. We don't know yet which gene, but hypothetically speaking, if there is a mutation, defect in synthesis of histidine, on the minimal media, which is this one, minimal media minus his, we will not see growth, but only it will grow in this media, which is minimal plus. Okay. This is the picture from another organism called Neurospora crassa. You can see what you will get. They did screen for adenine is another amino acid, leucine is another one. And now if you see, this is the minimal media. The mutant colonies are, let's say this is the minimal media with leucine and adenine. Everything is present in the, so yeast, the Neurospora, which is defective, they will not show defect because we are having leucine, adenine, everything is provided. It's like a complete media. So you see every colony we spotted is growing, the white ones. Now, this is the result of stamping. You came with stamp and you spotted it here, then there, then there. But this is minimal media where both leucine and adenine are missing. And you can see 1, 2, 5, 6, 7, 8, 9, 11, 13, 14, 15, 16, 17, 19, 20, and all of them are mutants. Because mutants in either leucine or adenine or both. And now if you go to if you bring in the minimal media leucine, you see it tells you that this one is defective in leucine because you brought back the leucine, it can synthesize, so it can now grow. So it is not defective in adenine because it can synthesize its adenine. And the only defect is leucine. The 5 is also leucine minus. If you now compare minimal plus adenine, so you can now very easily deduce, comparing with this one, which one is leucine minus, which one is adenine minus, and which ones are both. Okay, for example, these ones are not growing on when you bring back the both. So these ones are single mutants, and these ones, the last ones, are leucine minus, adenine minus, double mutants. Is it clear till now?
Yes, sir.
Yes, sir.
Yes,
Yes,
sir.
sir.
Now let's get into the practical examples. Let's say you ran the yeast mutant screen and you found, we go back to our original histidine screen, okay, this one. You found some mutants and you have to analyze whether your phenotypes are dominant or recessive. First thing, after your genetic screen, once you have mutant phenotypes, the first thing you analyze is it dominant or recessive? So what do? You simply cross met A his3 minus, its phenotype is his minus, we know already, with met A, with met alpha his3 minus, both are his3 minus, and then the expected phenotype is of course his minus, because this is also mutant allele, this is also mutant allele from the same mutant. These are, I told you, met A and met alpha, they will be haploid products, the gametes you create from your mutants. The diploid is written like this: met alpha slash A. This is diploid. But if you have written met A or met alpha alone, it means these are haploids. Now you cross your mutant met A, which is his minus, with met alpha but with wild type allele. In heterozygous state, if it becomes his plus, so what is your conclusion?
His plus is dominant.
Plus is
Sorry?
dominant. His plus is dominant.
It will be dominant.
So wild type is dominant, and what about your phenotype? His minus?
It's recessive, sir.
Recessive.
Recessive.
It's recessive.
Okay. So his 3 is recessive. So 3 means, let's say you did this screen. Imagine these are his minus mutants. This one, sorry. Now you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you number them: his 1, his 2, his 3, because these are independent colonies, independent mutants. So that's what the 3 is showing here. Okay. Now let's say one of your colleague went to another lab and they did a screen using yeast to discover heavy metal resistance or copper resistance. And here, if you pay attention, so their mutational assay will be different. You will have, for example, you mutagenized yeast and you spotted all your mutant population. Then you stamped it, and we call this stamping is called replica plating. Okay, replica, because this is exact replica of this. But here you have, growth media plus copper, and you want to see resistance acquired by yeast after mutagenesis. So it's basically you are going to see which of the colony shows growth in the presence of heavy metal or copper, which is not possible when all the others do not survive. They are dead. All of them are dead. Here, what was the case? Your mutants in the minimal media and the selection media, they did not survive. They only survived when you added required amino acid. Here, selection is like in the presence of copper, they survive. Now, again, you will do your cross, and your met A is resistant. So you call it cup 1R. R means resistant. this phenotype, you cross it with, let's say, MET alpha, and mutation is still resistant. This plus means wild type allele. CUP1 plus means wild type, and this one is resistant allele, which means minus. If you get 2N, the diploid is copper resistant, what is your conclusion about CUP1 minus?
It's dominant to.
It is dominant.
It's dominant to the wild type. Okay, so we have learned one thing so far. We have in humans also, we have, you know, natural alleles existing. For example, we have sickle cell anemia, which are, these are the mutations in hemoglobin gene, and in the hemoglobin gene HbS, S for sickle cell, if it is homozygous recessive, you know, they are defective in carrying oxygen and they are lethal. But natural, because, you know, populations, due to marriages in tribes, it's common in this heterozygous trait is common in, they discovered it in, I believe, in Africa, where they discovered that there are people who are resistant to malaria. And eventually they discovered that such individuals are actually heterozygous for sickle cell anemia. And the plasmodium, the mosquito bite and then transfer of the plasmodium, which causes malaria, it cannot infect these individuals. So this is one synthetic example that you created, you know, a heterozygous state, and due to mutation in this gene, organism has acquired in heterozygous state in the labs, you know, copper resistance. But this is a natural example where you see heterozygous state where HbS is actually acting as dominant, and, you know, individuals are acquiring a phenotype which is malarial resistance. Okay, now what is dominance and recessive? So far till today, we have talked about phenotypes as dominant and recessive. Dominant and recessive is phenotype, not genes. Please make it clear. Many people in their lives at very senior stage, they do make this mistake that, you know, this is a dominant gene, this is a recessive gene. It's a phenotype which is dominant or recessive. But we can try to explain what happens to a gene which gets mutated, that, you know, sometimes we see dominant phenotype and other times we see recessive phenotype. So this one, for example, is a gene X. So this is the wild type state, and gene gets transcribed, translated, and these are the protein products of the X. Now you mutated that, and due to a kind of mutation, it gets transcribed, translated, but almost very little amount of protein is available in the cell as compared to wild type. So this one we call loss of activity or completely absent, okay? But still something is being made. It may still perform function in the cell, okay? Now, such a state can be dominant, such a state can be recessive as well. It depends how important and crucial this protein product is within the cell. Because dominant or recessive means X plus over X minus. So one of the chromosome is like this, the other one is still producing 50% of the product. If, you know, 50% of the product is still performing function, it will most likely be recessive phenotype. But if 50% is not enough, it will be a dominant phenotype. We also call this loss of activity or loss of function mutation. The other state can be due to mutagenesis, you have a mutation in which there is hyperproductivity of this protein, and you see now much more amount of protein is there. We call this gain of new activity. So this protein, which is abundantly being produced as compared to wild type, this not only performs this function, but it acquires another function as well, and we call this gain of function mutation. Gain of function. And usually the dominant alleles are caused by increased activity, which is this one, or gain of or acquiring new activity. And recessive alleles maybe are due to partial loss of function. Partial loss of function. So let me let's take 5 minutes break here, because we are going to now go to the next stage of the genetic screen. We have covered that we created mutants, and after mutation we have determined whether it's dominant or recessive. So on my phone we have now 1531. Let's meet in it's 1532, so let's meet in 7 minutes from now, 1539. Okay.
Okay, sir.
Okay, so I'm back. Are you all back?
Yes, sir. Sir, can you please explain the
Yes, sir.
previous slide again?
Yeah, why not? This one. So here
Yes, sir.
yeah.
ये gene X or gene X.
So this is basically wild type, and this is mutant 1 in X, and this is another possibility in the same gene, mutant 2 in the same gene, individual two possibilities. And you know, in one case, so you can say this is X prime, this is X2 prime, allele of the X, okay? And due to mutation here, you have an effect of the mutation is, you know, hyper production of the same protein, and this protein is not only performing function for normal X, but it is doing function somewhere else of a totally different cell where normally it is not expressed or whatever. And it has acquired a new activity. And usually the dominant alleles are carrying such kind of behavior after mutagenesis. And recessive alleles are mostly partial loss of function or loss of functions, okay? Where you have, you know, in the presence of 50%, you here also you will have X2 prime over X plus, which means 50% is being produced. But since this has resulted in acquiring a new activity, so that is not caring about this 50%, it is now showing you a different cellular function as well, and the phenotype becomes more severe because this all protein is going somewhere else. Clear?
Yes, sir. So are you saying that gene X has two type of mutations in which there is a loss of activity and in another there is gene of, so there is increase in the
Good
activity.
question. So let's say here we are dealing with an organism, and all of them are leucine mutants. 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have, or since we are sticking to histidine, let's say we have histidine 1 mutant, histidine 2, histidine 3, so on, independent mutants. It could be this and this one carry mutation in the X, but this mutation is different than this mutation, and the end result is something like this. One mutant shows such phenotype, the other shows this kind of phenotype. Alleles after mutagenesis, alleles तो बहुत सारे बन सकते हैं ना, same gene hit हो सकता है कि नहीं हो सकता? It will become, it will become in, because we are going to continue this in this topic as well. So now what we have, just in response to the question she was asking, so let's say we have just touched upon what we are going to talk about now. We have discovered, you know, 10 mutants. Here's 1, here's 2, here's 3, here's 4, here's 5, here's 6, okay? And so on up to 10. Now the question is, hypothetically we said this could be a pathway for histidine, but it could be much longer pathway. Many more genes could be involved there. Now, how do we know that the mutants, 10 mutants we have discovered, are they in this gene, in this gene, in this one? Or since these are 10 mutants, it could be one mutant could be here, the other one could be in this gene, in this gene, in this, other genes in histidine. How do we confirm this? How can we solve this before we move on to map the gene and clone the genes? So we sort it out, we resolve this through classical genetic test, which is called complementation. So this is complementation. So complementation can only be done, this is very important, when two phenotypes are recessive. Remember, we are going to resolve whether these individual mutants, they are alleles of the same gene or they are mutations in two independent genes. In order to find this, we perform complementation test of our mutants. We take these 10, for example. And for complementation test, for example, we did this. We have his 3. We know his 3 is recessive, and remember, complementation is only possible, this test is only possible if we have recessive phenotypes. So let's say we have his X and his 3. We name them like this: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 10th is like his X. Now, one possibility is his X and his 3 are in the same gene. This is an independent mutation. This is an independent mutation. One possibility is his X is equal to his 3, which means, let's say this is his gene. After mutation, his 3, the one which we named his 3, it carries a mutation here. And his 10 or X in the same gene carries a mutation here. So now, if they are in the same gene, what do we call them? They are alleles of the same genes. Alleles. Or we will say these are allelic mutations. Allelic mutations. They are allelic to what? Same gene X, same gene which is history. So in this case, if we have his X is equal to his 3, that this will be the case. And if his X is not equal to his 3, when you will mate them, his X alpha mate to his X alpha, let's say, with his 3A mating type, the possibility will be now his 3 will be heterozygous and his X will be heterozygous in diploid, because they are in different mutation. So his 3 is contributing wild type copy of his X, and his X is contributing wild type copy of his 3. So the phenotype will be his plus. And what we will say? We will say his 3 and his X, they are complementing each other. Complementation. Here, his X and his 3, they are not complementing each other. Complementing what? His X mutant is providing wild type copy. Sorry. Here, his 3 mutant has provided wild type copy of his X, and his X mutant has provided his 3 wild type copy, because they are not in the same gene. They are not alleles of the same gene. They are non-allelic. So we call this complementation is taking place. Here we say they are allelic mutation, so there is no complementation. Let's say,
Sir, I didn't get this. Could you?
Yeah, please.
So this was kind of confusing. I didn't get it.
Yeah, it is. So I draw it here. Okay. We have, so is it clear up till now? We have 2 independent mutants, his 3 and his X.
Yes,
and
sir.
both show his minus phenotype, and these are recessive. It is clear until here?
Yes, sir.
Okay. Now, question is, is his 3 and his X mutants, are they both mutations in the same gene or in two independent genes? Is it possible they can be in two independent genes as well? Is it clear?
सर वो तो क्या एलिलिक हेट्रोजेनिटी नहीं होगी अगर डिफरेंट
hold on let me ask first the जिसने क्वेश्चन पूछा था कि क्लियर नहीं है, is it clear that they can be mutants in independent genes? So this his minus phenotype due to mutation in two totally independent gene in these two mutants. Is it possible?
Yes, sir.
Okay, now this is what we have to see, whether his 3 and his X, they are in the same gene or in the independent gene. If they are in the same gene, we will call them allelic. If they are in two independent genes, they are non-allelic. Now, what I showed you here is if his X and his 3 are in the same gene, but it will never be possible that the same nucleotide is mutated in both the alleles, his 3 and his X. There will be different nucleotides, mutation. So in this case, if they are mutation in the same gene, what will happen? Let's write them here. His 3 minus over his X minus. Now we are assuming that they are hitting, the mutation is in the gene, same gene, which I drew here. It could be here and it could be there. So what will be the phenotype? His
Minus, because both of them are his minus.
And since they are recessive, so both the copies are, though they are not same alleles, but both the copies of the same gene in the diploid, they are mutant. That's why we see recessive phenotype. However, if we have his 3 and his X, they are in two independent genes, and I try to draw independent genes. Let's say one is his gene, his 3 gene, and the his X is on totally different chromosome somewhere here. And independently, we already know independently they are recessive. Okay. Now, when we cross them together, what will happen? His 3 mutant copy will come from this one, but the wild type copy will be provided for his 3 by this mutant, gamete, the haploid of this. We will call this his 3 plus, this is minus, and let's put it semicolon to show you different chromosomes. This mutant is providing wild type copy of his X, this gene, and this one is having his X minus. What will be the phenotype of this diploid now? Will it be his minus or his plus?
It
is
should be
minus
जी आप बोलें दोबारा.
is it his minus
No, the gentleman who was talking to me who could not understand.
Sir, I think it should be his minus because 3 of the
No, it will not be his minus. Look, his minus can only be when this should be homozygous recessive or this should be homozygous recessive. This is what we saw here. Isn't true?
Yes, sir.
But in this diploid, it is heterozygous because they are not mutation in the same gene. Unlike this one, where they were mutation in the same gene, here they are mutation in two independent genes, which can contribute to histidine synthesis. And when they are two independent, so this one produce provided wild type copy of H3, and this one provided wild type copy of, sorry, this one provided wild type copy of his X. So they became heterozygous, and heterozygous states cannot be his minus, so that's why they will be his plus. Clear?
Yes, sir.
And
I
now
think
we
so.
call this, when this happens, we say complementation has taken place. So his X is complementing his 3 minus by providing wild type copy of his 3 plus, and his 3 is complementing his X by providing wild type copy of his X plus. Clear?
Yes, sir. I think so.
Okay.
Sir, where
you.
did the wild type copy come from if they're both like recessive?
Yeah, because we have haploids, the gametes, and there are 16 chromosomes. Let's say on one chromosome I said his X is lying and his 3 is lying on a different chromosome. When the two haploids mate in this diploid, now the wild type copy of his X from his X mutant wild type copy for his 3 from his X will come, and wild type copy of his X will come from his 3, and they complement each other. Is it clear?
Okay.
This will be clear here in the next example. Let's take now Drosophila example. We made a mutant and we were interested in wing development. When wings are defective, fly is paralyzed. So in German, this was done in a lab, I believe, in Germany. So they named these mutations the phenotype Shibere, SHI, this one. It's a homozygous recessive or dominant. They crossed it with wild type. They found all F1 flies were normal, which means this phenotype is recessive. In the same screen, they found, let's say, 10 different wing defective or paralyzed fly. Another independent mutant that was paralyzed minus minus, they crossed it with she minus minus to see whether now they are in, and paralyzed is also recessive. Okay, after such a cross, paralyzed minus over paralyzed minus crossed with wild type over wild type, the F1 was normal. So now they wanted to see if paralyzed and Shibere, they are alleles of the same gene. So same gene is defective, or they are in different genes. Now they crossed paralyzed homozygous recessive with Shibere homozygous recessive, and there are two possibilities. In one, let's say Shibere and paralyzed, they complement each other. What is your conclusion? It means Shibere and paralyzed, they are mutations in independent genes. They are not alleles of the same gene. For example, I write down here, this is fly chromosome. To make it simple, I assume we have X, Y. On second chromosome, assume we have paralyzed minus mutation. And on third chromosome, there is no mutation. Here, imagine we have female XX. On second chromosome, there is no mutation. And on third, we have Shibere, homozygous recessive. In F1, what we have here, what is written here, in principle, you are assuming something like this. I'm oversimplifying for you. Pi over plus, because wild type chromosome is coming from this side. And here, plus over Shibere. It becomes heterozygous. And in such a state, they are complementing each other because wild type copy for PAR is coming from this mutant in F1 and wild type copy of Shibare is coming from this mutant, and in F1 the fly will be wild type, normal. In such a case we will say this is the state that they are complementing each other. However, if F1 is also paralyzed, then it means they are not an independent mutation. They are mutation in the same gene, and there, what is the molecular thing? We have, for example, in F1, we have male or female, whatever, and then pair over Shibare, chromosome 3. It means it's actually at chromosomal level. If I draw this one at chromosomal level, the gene which carry mutation for pair and gene which carry mutation in Shibare, they are same. They are alleles of the same gene. They do not complement each other. Allelic mutations, they do not complement each other. In that case, you will say paralyzed and Shibare, they are equal. They are in the same gene. Is it clear until now?
Yes, but I have one question, sir, for like pair minus.
Yeah.
So in the second chromosome it's that, and in the third chromosome it's plus, right?
Yeah, yeah.
So what if in the third chromosome there's also the she, like, can that happen?
Absolutely. Very good you asked me. This was to explain you more simply. It could be both pair and she, they are on the same chromosome, but different genes. For example, pair is lying here, she is lying here on the third chromosome, but they are altogether different genes, altogether. And you will still get this result because they are non-allelic. They are not in the same gene. Same scenario which I explained you on independent chromosome, it can, and this is very much possible. It happens because the position of Shibare and pair, this is not controlled by us. ये तो nature ने design किया हुआ है ना? Is it clear?
Okay, so this test can still work for that.
Of
Also.
course, because they,
Okay.
yeah. Is it clear to everybody?
So they're going to recombine, right? If they're on the same gene, तो इसलिए जो है.
I did not get your question. Please repeat.
So I'm saying कि अभी the previous question was कि on the third gene, there can be a different genotype than what you made. So वो recombination से होगा ना? If they're present on the same gene, if the Shibare and the paralyzed gene
No,
are present
here
on the same chromosome, sorry.
Look, we are not talking about, यहाँ पे recombination का कोई role नहीं है. Forget about recombination.
Okay.
One parent contains Shibare. This hand where I have this gloves, it contains paralyzed. Gametes come in F1, okay? Even though they are on the same chromosome, but this black one is a different gene, this is a different gene. This gamete, the black one, brought in wild type copy for this gene. And this gamete brought wild type copy for this gene, although they are on the same chromosome. So they complement each other, and we will see wild type phenotype. The other possibility is they are not on the same chromosome, they are on different chromosome like this. And still they complement each other because they are in different genes, they're non-allelic. Is it clear? There's
Yes,
no
sir,
role
got
of
it.
recombination here.
Okay.
Let's go. If someone still hasn't understood, there's a third example now. Okay, so look, we started from yeast. His key examples आएंगे. Then I moved into fly, ताकि और clear हो जाए. अगर अभी भी understanding नहीं हो रही तो we have the third example now. Okay, again giving you the same example. What you have here is you are interested in flower color. Working in fly lab, a plant lab, and you are interested in development of flower color in this pea plant. You did mutagenesis, and in the many plants in the mutant population, you found many mutants. Mutant 1, mutant 2, mutant 3. So the wild type phenotype for color is purple. Okay? You cross your mutant 1 with purple, you get purple F1. So what is mutant 1 phenotype?
Recessive.
Right, so recessive. You crossed mutant 2 with wild type, it's purple. You cross mutant 3, it's purple, etc. So all are recessive. Now, the next thing you wonder if mutant 1, 2, and 3, are they mutations in the same gene? Are they alleles of the same genes? Or they are independent genes? You do complementation. And complementation test is possible because they are all recessive. So you cross mutant 1 with mutant 2. In F1, you get purple. White. So mutant 1 homozygous recessive crossed with homozygous recessive mutant 2. F1 comes white. You cross mutant 1 with mutant 3. F1 becomes purple. Now, here you are only adding phenotypes. White homozygous recessive, white homozygous recessive, F1 is white. Now let's put genotypes. Since they do not complement each other, you will say, okay, mutant 1 is homozygous recessive for a gene A1A1, homozygous recessive for the same gene because I get F1 white. So it's the same gene which carries mutation, but 2 means it's a different allele. That's why, so the A mutant in the F1, A minus, and F2 is also A minus, although this is allele 2, this is allele 1. So we will say A1 and A2, so mutant 1 and mutant 2, they are alleles of the same gene, or these mutations are allelic. Now, if you get purple, now you put phenotypes and you will say, okay, I know A1 genotype from here, A1, 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 but mutant 3 is not able to, or mutant 1 and mutant 2, they don't complement each other. Sorry, they complement each other. Here they don't complement each other. So since mutant 1 and mutant 3 are complementing each other, it means mutant 3 is mutation in totally different and independent gene in the flower color pathway, which is, let's say, B. So here, what is the genotype? A1, A, the wild type copy is coming from this side, the gametes, you know, and the B, which is coming from this side, and this gamete. So since the white phenotype is only possible when A is homozygous recessive or B is homozygous recessive, since it's purple, phenotype is not white because we have heterozygous. So now mutant 3 and mutant 1 are complementing each other. Complementation is taking place. And this heterozygous is giving you purple. Is it clear now?
सर
Yeah.
सर इसके अंदर हमने देखा है कि A1 के साथ जब दो डोमिनेंट बीज हो, सर बी भी कलर की उसका जीन कलर जीन है उसका.
Sorry, please repeat question.
सर जो म्यूटेंट स्ट्रेन 1 है उसमें A1 के साथ B
Yeah, because now they are not complementing, na? It's wild type. So
नहीं
you
सर, मैं वैसे ही पूछ रहा हूँ कि म्यूटेंट स्ट्रेन 1 में जो B है वो क्या चीज डिसाइड कर रहा है, जो सिर्फ B और B?
this one
जी सर,
capital B.
जी
Yeah. So this is now telling you that purple color is not only produced by A genes, there are other genes as well which are involved in pigmentation pathway. And if B becomes mutant, homozygous recessive, you still can get white phenotype. But when you are getting purple, to solve this one, this is a dihybrid state now, independent genes. So you are trying to solve, when I carry homozygous recessive, I write it here, when I carry homozygous recessive A1 cross to homozygous recessive B1, how can I get purple phenotype, this one? Because this one tells you that they are no more homozygous recessive. They are not this state. They are not allelic. A1 is not equal to B1. So you say, okay, complementation is taking place and the wild type allele of capital B is coming from this parent, and wild type copy of A is actually coming from this parent, because P has many genes, and pigmentation pathway, just like amino acid biosynthetic pathway, it can be relying on 10, 20, 50 genes. Now it means the purple became like this. That's why we have this purple, because pathway for pigmentation is getting wild type copy of A, wild type copy of B, so complementation is taking place. Clear?
सर मैं वैसे पता पूछा था कि अगर A और B में से दोनों में कोई एक भी होमोजाइगस रिसेसिव हो जाए तो वाइट ही कलर शो होगा, पर्पल नहीं होगा.
Okay, so you are asking if one of the two, for example, if AA is homozygous recessive, it can give white. Yes, it is giving you white. If B is giving you homozygous recessive, it can give white. Yes, it can give white. Now your question is, sir, is it possible if I have this state, homozygous recessive, and BB, can it be purple? This is another
sir.
possibility.
Okay.
Clear?
Okay.
This is the question?
Yes, sir.
Yes. We will solve this in next lecture when we will talk about non-allelic interactions, which are called epistasis. When two genes, A and B, they are interacting with each other in the same pathway. So after this complementation, the next thing is to see if they are, how they interact with each other. The answer to your question will be in next one. Okay, now let's, the last few minutes. Complementation experiment, for example, we did in yeast. We also looked at flies. We looked at now the plants. This is called complementation table. Let's say you have now 5 different or 6 different white phenotypes, and you do all these complementation crosses. What you see here, you draw a table, mutant 1 cross to mutant 2. What is the phenotype? Purple. Mutant 1 cross to mutant 3, purple. Mutant 1 cross to mutant 4, purple. Mutant 1 cross to mutant 5, white. Mutant 1 cross to mutant 6, purple. So you draw such a table. Then mutant 2, of course, mutant 2, you cross to mutant 2, you know it is bite, you know, it's a homozygous recessive. But mutant 2, when crossed to mutant 4 or mutant 6, it gives you white, but not purple here. But purple is only when you have mutant 2 crossed to mutant 3 and mutant 5. Now, please answer my question. If you look at mutant 1 in relation to all these mutants, where mutant 1 is complementing and where complementation is not taking place?
It's not taking place in mutant 5.
Very good.
But in the other, all the other ones, it is complementary.
Very good. Okay. So what is mutant 1 and mutant 5? X1, X1 is equal to X5, X5. These are alleles of the same gene. Clear?
Yes, sir.
Yes.
And X1, X2, X1, X3, X1, X4, X1, X6, these are not alleles of the same gene. X1, X1, these are not alleles of 2, 3, 4. They are independent genes. However, X2 and X4, X5, what are they?
So X2 and X4 are non-complementary, and X2 and X5 are complementary. X2, X4 lie on the same gene.
X2, X4 and X2,
Are
X6.
not. X2, X4 and X2, X6 are not complementary. They all lie on the same gene.
Yes, they are allelic. Okay, they are alleles of the same gene. Very good. And X2, X3 and X2, X5.
They are on different genes.
Yes, they are on different gene. These are non-allelic. They are independent genes. So there's a tricky term here now. Don't be confused. Every year students get confused, so don't worry. When complementation takes place, which means X1 and X2, they're complementing each other, it means different genes are mutated. It's simple, very simple. When there's no complementation between, for example, X1 and X5, lack of complementation, it means alleles of the same gene. So far, so good, simple. Now, after this table, the ones which are not complementing each other, for example, X1 and X5, they are not complementing each other. You will call this complementing group, complementation group. It's slightly confusing. You would say, no, sir, we should say this one complementation group. No, in genetics, complementation group is the ones which do not complement each other. Alleles of the same gene, which means these ones where lack of complementation, we will call them complementation group. And how we represent them? We had how many? We had X1, 2, 3, 4, 5, 6. Now, this whole table can be summarized like this. This table is summarized like this. In a circle, you draw X1, 2, 3, 4, 5, 6. Now, the ones which are connected with one another, these ones, for example, X1 and X5, these are complementation group. Complementation group. And what we just learned, the non-complementing, where lack of complementation is this one, X1 and X5, they are connected. The ones which are connected with one another, it means they are alleles of the same gene. They are alleles of the same gene. Similarly, 2, 4, and 6, 2, 4, and 6, they are alleles of the same gene, so you connect them with each other and it becomes a triangle. So 2, 4, and 6 is another complementation group, which means non-complementing and gene, what I say, the alleles of the same gene. So 2, 4, and 6 are alleles of the same gene. The 3, it does not show any of the wild phenotype. So we say X3 complements all the other alleles. It represents another gene. So based on this test, how many genes we have discovered so far? How many genes we have discovered? How many independent genes we have discovered which can cause white phenotype? Hello? How many? और कोई?
So I think 4, 1, 5, 4, and 6.
1, 5,
4 and 6.
Who said 3?
Sir,
I. Who is I? Sadaf. Sadaf is right. So look, 1 and 5 are allele of the same gene, complementation group. X2, 4, and 6, alleles of the same gene. And X3, which complements all the others. So they are one independent gene. They are alleles of the same gene. They don't complement them and they don't complement them either. And this one is, so we have discovered 3 independent genes. But for this gene, we have 3 independent alleles. For this gene, we have 2 independent alleles. And for this gene, we have only 1 independent allele. Clear until now? And this is the molecular level. Go back today. This is the last slide. And this is what is happening. When you have hypothetical scenario, if X1 and X2 are mutation in the same gene, this will be the case. They will fail to complement each other and we will get white flowers. If X1 and X2 are 2 independent genes, now X1, the wild type copy will be provided by from this parent X2, and the wild type copy for X2 is provided by this parent through its gamete, and complementation does not take place because we have wild type protein being made 50% from this wild type copy, and recessiveness disappears and we get purple flower. And we say this is complementation is taking place. This is at the, so next week when we, next week or next lecture, today is the last lecture of the today's week, this one.
Yes, sir.
Yeah, so we will talk about non-allelic interactions, and non-allelic interaction means we will start from here. Now once we have non-allelic means these are 3 independent genes, and if all 3 are playing a role in purple pigmentation, how they 1, 2, and 3, how they interact with each other, how they made purple pigmentation possible. Clear, Ji?
Yes.
So I have a question.
Ji, please.
So can we say that if 2 different mutations are happening at the same locus, then it will lead to the same phenotype?
Yes, same locus means same gene, but different nucleotides. It can give you same phenotype. This is what is happening here. This X11, okay, go back here. This one. Same locus, but different positions maybe in the same gene. Anyone else? Any other question, please? Hanji, no more questions? Today was the 4th lecture. Next one is the last, and then we will have an exam. So far I'm hearing that you are doing good in assignments. So these assignments are very crucial. That's why, we gave you these small, and these are not tricky, I would say these are not complicated things. These are more analytical, and I want your brains to work as much as possible so that when you see these things in exam, you're not simply blown away, from where these numbers are coming. We have pretty much covered everything for the first module except the next lecture, which is the epistasis, which is non-allelic interactions. It's one of my favorite module in this whole course, because people 100 years ago, they were doing these mutant screens in different organisms, fruit fly and yeast, and then they were mapping chromosomes and doing these simple genetic analysis, and based on that they were predicting the biochemical pathways. They were super genius people. No more questions? Okay, then. Thank you very much. I wish you a
Thank you, sir.
nice weekend.
Thank you,
and
sir.
good
Love
with your the rest of the lectures. Allah
Thank you,
Hafiz.
sir.
Thank you, sir.